Khalid has leveled the whole field with his prowess of intelligence as he tries creating a soup of revolving nutrients, kind courtesy the sun, through electrochemistry. These past few days he has been living like he used to before he successfully made his plantation, which came to a tragic end not long ago. He had made his plans straight. He arranged his wires and made sure all the equipment he needed were available. Of course not. He didn't, as the circuitries were not going to be sufficient due to the insufficiency in silicon materials. But his immense knowledge in optoelectronics kept him to work, grinding and chipping, mixing some of these materials, chippings, and sand particles with the help of the sword he has. From the look of his plan, he had a long way to go, but he had a dream and nothing was stopping him. Light's a very dependable source of energy. Just like Khalid's whole plan stemming from solar energy, plants and even life's whole existence stems from solar energy. In a process known as photosynthesis, we have the light-dependent reactions and light-independent reactions. Photosynthesis is the reverse of respiration. And let's see. Respiration is the process by which cells obtain energy by consuming and breaking down oxygen and the release of carbon dioxide for the production of ATP. Plants or autotrophs in general convert light energy into chemical energy, and this is the process of photosynthesis. Basically, photosynthesis combines carbon dioxide and water into glucose and oxygen gas, whereas respiration does the exact opposite. When we talk about photosynthesis, photo means light and synthesis means creation. So this is the creation of something using light. Photons from the sun are absorbed by a plant pigment named chlorophyll. The thing that plant cells have that animal cells do not are plastids. A most important plastid is the chloroplast. The chloroplast is a structure containing the pigment called chlorophyll. The thylakoid is also a structure that contains this chlorophyll, and a stack of these thylakoids is known as the granule. Multiple thylakoid stacks are called grana. Inside the thylakoid is the lumen, and outside the thylakoid is the stroma, and the stroma is just confined within the chloroplast. Light-dependent reactions occur in the thylakoids, and the light-independent reactions occur in the stroma. Thylakoid membranes are phospholipid bilayers, and this means they are very good at maintaining concentration gradients of ions and proteins. Now let's delve into the light-dependent stage. A photon enters into a molecule of chlorophyll, and this starts the process of photosynthesis. An electron inside the chlorophyll absorbs the energy, thus gets excited. An energized electron or excited electron is carried away from the chlorophyll by a mobile electron carrier or a plastoquinone. When this process happens, the chlorophyll splits nearby water molecules, which originated from the roots, into hydrogen and oxygen molecules so as to replenish the electron that it has lost with the hydrogen. This is a reduction reaction of the water into oxygen molecule. This first chlorophyll is part of a complex of proteins and lipids called the photosystem 2 or plastoquinone oxidoreductase. From this point, an electron transport chain is the chain this electron will be going through, losing its energy in the remaining stages there is. The second protein complex is known as the cytochrome complex, and here the cytochrome serves as an intermediary between photosystem 2 and photosystem 1. and it uses a little bit of the energy from the electron to pump a proton from the stroma into the thylakoid, that's the lumen. So the thylakoid starts getting filled up by protons, and as the proton concentration inside the lumen increases, the proton concentration in the stroma decreases, creating a concentration gradient. Since the protons are now more abundant in the lumen than in the stroma, the enzyme ATP synthase uses these protons to combine the ADP and phosphate into ATP. ADP being adenosine diphosphate and ATP being adenosine triphosphate. This enzyme packs an inorganic phosphate onto an ADP, making ATP, that's cellular energy, actual energy, due to the concentration gradient. Then the electron now moves into photosystem 1, where the electron is hit with another photon, and this time all of its energy would be used in making NADPH, and that's nicotinamide adenine dinucleotide phosphate. This is also an energy container just like the ATP. This is how it happens. Another enzyme, NADPH synthase, is also available to combine 2 electrons and 1 hydrogen ion with something called NADP+, and these are essential for energy conversions. So the combination of these 3, that's the NADP+, the hydrogen ion, and 2 electrons, forms NADPH. So after the light dependent reaction, we are left with the nicotinamide adenine dinucleotide phosphate reductase, NADPH, and adenosine triphosphate, or ATPs. Now we move to the Calvin cycle, which is the light independent aspect of photosynthesis. This begins in the stroma. The first phase is known as the carbon fixation phase, where a CO2 molecule, as carbon dioxide, is fixated onto a ribulose biphosphate by the help of an enzyme known as ribulose 1,5-biphosphate carboxylase oxidase, or Ribisco for short. This combination creates 2 molecules of 3-phosphoglycerate, or 3GP. Phosphoglycerate kinase is powered by some ATP, which joins the phosphate groups onto phosphoglycerate by reducing the ATP into ADP and phosphate. And this phosphate is transferred by the phosphoglycerate kinase to produce 1,3-biphosphoglycerate. 3GP dehydrogenase removes some electrons from NADPH, reducing it into NADP+. This gives the combination biphosphoglycerate some electrons, and then we get 2 molecules of glyceraldehyde 3-phosphate. This is a high energy compound that plants could convert into any carbon like glucose, cellulose, starch. Because of this, 3GP is considered the ultimate product of photosynthesis. 3GP are needed to regenerate the ribulose biphosphate, and so this process goes on until 5 3GPs are formed, where 1 molecule of 3GP leaving the other, so it could be used or converted into ribulose biphosphate, and the process goes on again in a cycle. So the Calvin cycle converts 3 molecules of CO2 into 1 molecule of 3GP, and it requires the use of 9 ATP molecules and 6 NADPH molecules. So if 1 molecule of glucose needs to be produced, 6 molecules of CO2 is needed. Khalid has been working tirelessly day and night on this chipping and these circuits he was making, sharpening the tip of the edge always so he could use it as a soldering bit of sorts, placing it in fire mildly and soldering these components to the boards. He was getting nowhere. But he looked up. He heard something approaching. He looked from the dust and he saw a figure.